Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about the cell cycle and its regulation. Cell cycle means what? Cell cycle means the overall events that occur during the division of one cell to form two daughter cells, or the stages through which a cell passes from one division to the next. During this phase, cell grows and prepares itself for the division. So, at first, cell prepares itself for the division. Next, it actually divides. So, the overall cell cycle is divided into one interphase and then M phase. Actually, the division phase is very small or for a short duration, but the preparatory phase is very long and called as interphase. Interphase, where the cell prepares itself for the upcoming division, and the M phase, where the cell actually divides. Okay. So the whole cycle, in whole cell cycle, at first the DNA doubles itself in the S phase, and next half itself in M phase. During anaphase, half of the chromosome move to one pole and half of the chromosome move to another pole. So it occur in anaphase of the M phase. Next is cell cycle, which completes in two phases. The first phase is the preparatory phase, where the cell prepares itself for the upcoming mitosis process. So interphase is again subdivided into three phases. One is G1 phase or gap 1 phase. Second is S phase or synthesis phase. Then G2 or gap 2 phase, leading to doubling of genome. Actually, in S phase, the DNA replication occur and DNA duplication occur. Next is the M phase, where actual cell division occur. So M phase is the phase of division and divided into two subphases. One is karyokinesis or nuclear division, which is again divided into four subphases. The first phase is prophase, then metaphase, then anaphase, then finally telophase. After karyokinesis or nucleus division, the division of cytoplasm occur, which is called as cytokinesis. So the M phase is divided into karyokinesis and cytokinesis. Okay, and this cytokinesis is the division of cytoplasm, leading to having of genome passing into daughter cells. So at first this is one cell. At first the nucleus divides into two. Next, the cytoplasm divides, forming two daughter cells. After the cytokinesis, cytoplasm divides, forming two daughter cells from one mother cell. Okay. Next, this is the diagrammatic representation of the cell cycle at first interphase, which is divided into G1, then S, then G2. Next is the M phase, leading to the formation of daughter cells. Sometimes after G1, the cell thought to rest for some time or arrest the cell for some time and do not enter into G1, S or G2, and that phase is called as G0 phase or gap 0 phase or quiescent stage. So at that stage, no cell division or no event occur inside the cell. Okay. Next is the events occurring in G1 phase or gap 1 phase. The events occur, that is synthesis of enzymes which are required for DNA replication, because DNA replication is to occur in S phase. So cell prepares itself for the upcoming DNA replication. So at first it synthesizes all the enzymes required during the replication. Next, the RNA needed for transcription and translation. Next, the synthesis of adenosine triphosphate or ATP to provide energy for cell division. Next is the synthesis of raw material required during the DNA duplication. So one is pentose sugar, then phosphoric acid, then nitrogenase, it is not nitrogenase, it is nitrogen bases, purine, pyrimidine. Okay, adenosine, guanosine, then cytosine, okay, thiamine, these are various nitrogenous bases. These are required during the DNA duplication or replication in S phase. So these are formed in G1 phase. So many things are synthesized in this phase. Therefore, the size of the cell finally increases. Okay. Next is the S phase. The events occurring in S phase. The most important thing is DNA replication occur in S phase. Next is centriole divides, but only in case of animals, because plant cell lacks centriole. And synthesis of histone protein is, it is not histone, it is histone protein, which is required for packaging of DNA. Now the cell is ready to enter the next G2 phase or gap 2 phase. Next, events occurring in G2 phase. Synthesis of tubulin protein required for spindle formation. Spindles are required during anaphase to pull all the half of the chromosome to one pole. Next is the synthesis of proteins which are required for plasma membrane formation. Next, cell organelles are doubled for daughter cells. Lots of ATP molecules are required for movement of chromosome from equator to pole. Approximately 30 ATPs are formed, so ATP synthesis increases. RNA synthesis takes place. Now the cell is ready to enter the next phase, that is M phase or mitotic phase. So events occurring in M phase, at first karyokinesis, because M phase is again subdivided into two phases. One is karyokinesis, second one is cytokinesis. So karyokinesis again subdivided into four phases. One is prophase, where chromatid coiling occur. Next, disintegration of nuclear membrane and nucleolus, and spindle formation started. Next is the metaphase, where chromosomes arranged on the equatorial plane. Next is anaphase, where movement of chromatids occur towards the opposite poles. And next and final stage is the reconstruction of daughter nuclei. And next phase is the cytokinesis, where the cell plate is formed leading to equal division of cytoplasm. Cell plate is formed in case of plant cell. In case of animal cell, cell furrows are formed leading to formation of two daughter cells. Next, so these are the diagrammatic representation of different stages of the cell cycle. At first the interphase, then in prophase the chromatin coiling occur forming systematic chromosomes. Next they are arranged in equatorial plane. In metaphase, next half of the chromosome move to one pole, another half move to another pole. Next is the telophase, where the cell plate formation started. Next the cytokinesis, where the cytoplasm divides to forming two daughter cells. Next, after cell division, each of the daughter nuclei begins the interphase of a new cycle. Okay, but some cell, after remaining in G1 phase for some times, come out of the cell cycle and enter to G0 phase, that I have already discussed about the quiescent phase. In quiescent phase, the cell division stops, but other activities of the cell continues. The cells cells of heart cell, then kidney cell, liver cell, neurons, sometimes they undergo G0 phase. They cannot divide in G0 phase, that is a resting phase. Sometimes the cell re-enters the cell cycle from quiescent phase then when required. For example, during the formation of periderm, during secondary growth. So this graph shows the cell cycle of a cell showing changes in the DNA content during various phases. In G1 phase it is 2C, and during S phase the amount of DNA gets doubled. Then in G2 phase we have increased amount of DNA. Then in N phase again it gets lowered. Then in G1 phase again the amount gets lowered. So in this way the DNA content changes in various phases of cell cycle. Next, regulation of cell cycle. Cell cycle does not occur in unchecked manner. It occurs in a very controlled manner. The preparations of the cell are checked by regulatory molecules. It includes detection and then repair of genetic damage as well as prevention of uncontrolled cell division. So there are two classes of regulatory molecules. One is cyclins, other one is cyclin dependent kinase or CDKs. So the Nobel Prize in Physiology and Medicine in 2001 was awarded jointly to Leland H. Hartwell, Tim Hunt and Sir Paul M. Nurse for their discoveries of key regulators of cell cycles. So they discovered the regulators of cell cycle, which regulates the cell cycle or controls the cell cycle. Okay, next the regulatory molecules, cyclins. These are D-cyclin or G1 cyclin, then S-phase cyclin, which is cyclin E and A, then M-phase cyclin, B cyclins. Their levels in the cell rise and fall with different stages of cell cycle. Next is cyclin dependent kinase. One is G1 cyclin dependent kinase. The name of it is CDK4. Next is S-phase CDK or cyclin dependent kinase CDK2. M-phase CDK CDK1. Their level in the cell remains stable. And this is the difference between your cyclin and cyclin dependent kinase. But they remain inactive stage. They remain in inactive stage. They bind to appropriate cyclin in order to be activated. Okay. Next, their function is to provide phosphate group to a number of proteins that control the process of cell cycle. So, kinases are employed to provide phosphate group to cyclins. Okay. Next, these are different regulatory molecules. The name of cyclins like cyclin D, which is associated with CDK4. Helps in G1 phase to inhibit RB protein and signal the cell to prepare the chromosome for replication. Next, S phase cyclin, then G2 phase cyclins. S phase cyclins are cyclin A and E and CDK2, which activates the DNA replication to occur. Next, G2 phase, in G2 phase cyclin B and CDK1 will act to activate mitosis. Next, cell cycle checkpoints. We have three main checkpoints. One is between G1 and S, which is present before the cell enters into S phase. Next, G2 to M checkpoint after S phase. Next is APCC checkpoint during mitosis. Okay. So at first G1S checkpoint, before the cell enters into the S phase, this checkpoint checks for the cell size, because during G1 phase the size of the cell increases. Next is it checks for nutrients, then check for DNA damage, check for all the preparations, checks whether the S phase cyclins and CDK complex is activated or not to initiate the upcoming DNA replication. When cell completes or fulfills all these criteria, they can move to the next phase, that is S phase. So these are like traffic post, okay, traffic police. Next, G2M checkpoint, which is present after S phase. Here it checks for proper DNA replication, checks for all preparation for M phase, checks for tubulin synthesis. It checks whether the M phase cyclins and CDK complex is activated or not to initiate mitosis. When it completes all the required criteria, it can pass to the next phase, that is M phase. So all these checkpoints are required require the service of a complex of proteins. The level of these proteins are increased in damaged cells. They allow time to repair DNA by blocking the cell cycle. P53 is one such protein which senses the DNA or detect the DNA damage and can halt the progression of the cell cycle in G1 phase by blocking the activity of CDK2 until the damage can be repaired. If the damage is so severe that it cannot be repaired, then the cell destructs itself by apoptosis or programmed cell death, where the cell destructs itself whenever it detects any mistake or any damage. In case of the damage to DNA after S phase, the action of CDK1 is inhibited, thus stopping the progression of cell from G2 to mitosis. So in this way, different checkpoints and different protein molecules are acting to control different processes, to detect different damages. So it is all about your cell cycle, different stages of cell cycle, about different checkpoints. In a very short time, I have tried to discuss all the things bit by bit. So please appreciate my hard work. Please like my videos. Please share with your other friends. And please subscribe to my channel Sunanda's Tutorial. Thank you.